Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science.
If you appreciate the deep dives we take, please take a moment to like, subscribe, and share with your friends and family. It really supports the Sand Science Experience.
Today, we are doing a deep dive into what is really the cutting edge of longevity research.
That's right. For centuries, we've thought about aging just by the calendar, a simple chronological number.
But biologically, that number can be, well, pretty meaningless.
We all know people who are 60 but seem like they're 40, and the other way around.
Right, and that's because aging is what they call heterogeneous. It's not just that it happens at a different pace between people.
It also happens at a different pace inside one person. Your liver could be aging faster than your lungs, for instance.
So that kind of complexity, it really demands a new way to measure it, a molecular way.
Exactly. If you want to actually understand and maybe intervene in the aging process, you need these systemic clocks that can measure the biological age of individual organs.
And that's our mission today. We're unpacking a huge study that just came out November 2025 in Nature Aging.
And this team used some really advanced tech, plasma proteomics and machine learning to build these personalized, organ-specific aging clocks.
Plasma proteomics sounds complicated, but it's basically just measuring thousands of different proteins floating around in your blood, right?
That's all it is. And those proteins are like messengers. They carry information about what's happening in every single one of your organs.
And I think the key idea for you, the listener, is that we're moving beyond that sort of general question, how old are you?
How old are you in general?
Yeah, to something much more specific. How old is your brain, really? Or your heart?
That's the level of personalization this research unlocked. They actually developed 10 specific organ clocks.
10? So which ones?
Brain, heart, lung, immune system, artery, intestine, liver, kidney, muscle, and pancreas. Plus a sort of overall clock for the whole organism.
And the main concept they used to see if you were aging well or not was this idea of an age gap.
Exactly. The age gap is the central metric here. It's pretty simple in concept. They take your blood, measure the proteins, and a model predicts your proteomic age.
And if that predicted age is higher than your actual age...
You've got a positive age gap, accelerated aging. If it's lower, you've got delayed or, youthful aging compared to your peers.
Okay, so let's get into the foundation of this. If you're going to build 11 different clocks, the data you train them on has to be just immense, right?
It was massive. They built the clocks using the UK Biobank data set. We're talking about 43,616 people.
Wow.
And that's just the training ground. The really critical part is validation.
Right, because a clock that only works on one specific group of people isn't very useful.
Not at all. So they tested them rigorously on two completely different groups, the China Kadoorie Biobank with almost 4,000 people, and the US-based Nurses' Health Study. So you have this great diversity in ethnicity and geography.
And how well did they work? When you test them across these different populations, did the clocks hold up?
They held up extremely well. The correlations were incredibly high, with R values up to 0.98.
And an R value of 1.0 would be a perfect match, for context.
Precisely. So numbers like 0.98, 0.93, that means these clocks are incredibly reliable no matter your background, a really robust biomarker.
But being reliable is one thing. Being specific is another. How did they prove these were actually 10 different clocks and not just, one general aging signal measured 10 ways?
That's maybe the most fascinating part. First, they found proteins that were specifically enriched in each organ, like 117 specific proteins for the brain clock.
And then they compared the age gaps. So if my heart is aging 5 years too fast, does that mean my kidney is too?
Generally, no. Very little, in fact. And they found the correlations between the different organ age gaps were surprisingly weak.
So that confirms it. Your heart can genuinely be older than your lungs.
It absolutely can. These clocks are capturing unique organ-intrinsic aging.
So here's where it gets really interesting for me. Even with all these distinct clocks, did they find any organs whose aging was sort of more central to your overall health?
Yes. Despite having very little protein overlap with the main organismal clock, two systems really stood out.
And they were?
The brain and the arteries.
Wait, that seems counterintuitive. If they're so central to overall aging, why would they share so few proteins with the overall clock? Only 6% for the brain, you said.
It is, and it means we might need to shift our thinking. It suggests these two organs could be the primary drivers, or maybe the most sensitive canaries in the coal mine.
So their failure predicts a system-wide failure, even if the molecular signals they give off are unique.
Exactly. A problem in the brain or the vascular system just cascades through the rest of the body.
Which brings us right to the so what question. This isn't just an abstract number, it has real predictive power. What did they find about predicting actual disease?
The power was, frankly, startling. They found an accelerated age gap in an organ strongly predicted future disease and even all-cause mortality.
And this was on top of the usual risk factors.
Completely independent. It didn't matter what your BMI was, your cholesterol, or even your known genetic risks. This was a separate, powerful signal.
Let's focus on the biggest one, mortality. Which organ clock was the biggest warning sign?
The brain clock was the clear leader. For every 1 standard deviation increase in your brain age gap, your mortality risk went up by a hazard ratio of 1.44.
Can you put that 1.44 number in context for us? What does that feel like?
Sure. A 1 standard deviation increase means your brain is aging faster than about 84% of your peers. And that 1.44 means your risk of death from any cause is 44% higher.
Just from a blood test, that's a huge signal.
A massive signal. And for specific diseases, the links were even stronger, especially with neurodegeneration.
I would guess so.
Yeah, for all-cause dementia, the hazard ratio is 1.88. So an 88% increased risk. Multiple sclerosis was 1.52. Parkinson's was 1.38. Very strong connections.
What about diseases outside the brain, like diabetes or kidney disease?
This was another surprise. The kidney and the intestine clocks were the strongest predictors there. For chronic kidney disease, the intestine clock was actually a stronger predictor than the kidney clock itself.
The intestine predicts kidney disease. That just hammers home how interconnected everything is.
It really does. And they had a perfect example of this with Alzheimer's patients. They looked at their blood samples from years before they were diagnosed.
And what did they see?
At baseline, these people had a brain age gap of, on average, 2.2 years of acceleration.
Okay.
But their overall organismal age gap was only 0.5 years. So the brain was screaming a warning signal that the general body clock was completely missing.
That right there validates the whole organ-specific approach.
Absolutely. And they formalized this by creating what they call extreme agiotypes.
What does that mean?
They looked at people who had 5 or more organs in that extremely aged category. A clear multi-organ failure pattern.
And their risk.
It was staggering. A 7.8-fold higher risk of mortality.
Wow. Nearly 8 times the risk.
But on the flip side, people with 5 or more extremely youthful organs, the super useful agiotype, had a 60% lower risk of death.
It's clearly additive then. The more youthful organs you have, the better.
It's a very clear dose-dependent effect.
Let's circle back to the brain for a minute. Since that clock was so predictive, what proteins were actually driving it? Do we know what's going on molecularly?
Yes, and this is the beautiful part of proteomics. The proteins tell a story. The top ones linked to dementia were things like GFAP.
Which is a marker for what?
Reactive astrocytes. Basically, it's a sign of inflammation in the brain's support cells.
Okay, so inflammation. What else?
There was NEFL, which is a marker for neuroaxonal injury, direct damage to the nerve fibers. It's so important it's already used in some clinical trials.
It's damage to the brain's wiring.
Exactly. And others pointed to problems with synapses, the connections between neurons and vascular dysfunction. It's a very clear picture of what's breaking down.
It turns the clock from just a number into a diagnostic profile.
Precisely. And then they took it one step further and looked at genetics.
Right, the APOE4 gene is the big one for Alzheimer's risk. How did that play into this?
This might be the most hopeful finding in the whole paper. They found that the brain age gap predicted Alzheimer's risk, even after you accounted for your APOE4 status and your overall genetic risk score.
So it's an independent risk factor?
It's independent, but it's more than that. It seems to be protective. Individuals who had the high-risk APOE4 gene, but who also had a super youthful brain age, showed a significantly reduced risk of dementia.
So a biologically young brain can actually override a bad genetic hand.
It appears so. It suggests that your genetics might load the gun, but it's your biological age, which is modifiable, that pulls the trigger.
That's a huge takeaway. Which brings us to the modifiable part. What did the study find about lifestyle, the things we can actually control?
The links were very clear. Unhealthy behaviors, especially being sedentary, a poor diet, smoking, were tied to accelerated aging, particularly in the brain and pancreas.
And on the positive side?
Healthy lifestyles, physical activity, good sleep were linked to slower aging. Again, especially in the brain and the intestine. It shows these interventions have real, measurable, organ-specific effects.
This is all amazing research, but how do you get this into a clinic? You can't run a test that measures thousands of proteins for every patient. Did they address that?
They did, and it's a crucial step. They used a machine learning method to basically strip away the non-essential proteins to find the smallest possible panel that still works.
And how small could they get it?
For the main organismal clock, they got it down from 240 proteins to just 20.
Wow, just 20 and the accuracy.
It still retained over 88% of the original performance. That's a huge simplification that makes it clinically feasible.
And what about the all-important brain clock?
They did even better there, in a way. They refined it from 70 proteins down to a super-focused 10-protein model.
10 proteins?
10 proteins. And it still held on to over 88% of its predictive power. And that is a simple, non-fasting blood test that could track your risk for neurodegeneration.
So, to sum this all up, what these clocks give us is a real molecular reason for why we all age so differently. And they're tools to track the health of our organs years before we'd ever get a diagnosis.
It's a roadmap, a biologically interpretable roadmap for tracking health. And by seeing which proteins are going wrong, synaptic loss, vascular dysfunction, we get these incredibly specific targets for new interventions.
So let me leave you with a final thought to mull over. If having a super youthful brain can potentially override a major genetic risk like APOE4, how soon will it be until we have interventions, lifestyle, or even drugs that specifically target those 10 brain proteins, personalized directly from your organ-specific age gap?
That level of precision is now within reach. It shifts the entire goal from treating disease late to preventing failure early.
And that wraps up this deep dive into the forefront of aging science. Remember to like and subscribe to support the Sand Science experience.
We'll catch you next time.